Hi Eleven, we'll try this. I'm trying to record on a PowerPoint. We'll see if this is any better. Right, so this lesson is on transpiration and translocation. So two really similar words that are actually quite different. So I'll put the definition on there if you want to pause them now and try and understand this, but obviously that's what the lesson's on, so these words will make more sense soon. Right, so they both start with trans, which means across or through. So the movement of things in plants, and transpiration is the movement of water, really importantly. So transpiration, and a link you can make there is, so sweating is perspiration. So perspiration, you can think of like water, sweat, water. Yeah, so transpiration is a movement of water. Translocation is the movement of sugars, which is mainly sucrose, through the plant. Right. Before we understand transpiration, we're going to do first the movement of water. We need to understand what a water molecule would look like. So it's H2O, and it's a covalent bond between a hydrogen and an oxygen. How many electrons does hydrogen have? It's got one. It wants two to have a full first outer shell. And what it does is it does a covalent bond with the oxygen. So what we can think of is, so the hydrogen nucleus is here with a proton in, and here with a proton in, and its electrons are kind of caught up on one side of it. So what that means is the hydrogen has a slightly more positive side because of this proton. So if you do A-level biology, you say this is delta positive. It's basically slightly more positive than the rest of the atom. So part of the atom is positive, and part of the atom is negative because the oxygen has electrons on its outer shell that aren't getting shared with the hydrogen. So this side of the cell, because of these electrons, the electrons are negative. So this bit is slightly negative, delta negative. So we can think, forget about delta if you want, but part of the molecule is slightly negative, part of it's positive. So it acts like a little magnet. It's a polar molecule, we'll call this. So what happens is water attracts other water molecules. So if we've got a water molecule here, the positive bit will attract the negative bit. The negative bit of that will also attract a different positive bit. So water is a sticky substance. You don't really think about this, but it is. Like water hangs around in droplets. Like, why does that not just split apart? It's because it's got these forces, which we call intermolecular forces, so it sticks together, right? And that's going to be important to understand the rest of transpiration. So water is a sticky molecule. Why is it sticky? Because it has slightly positive bits and slightly negative bits. Why does it have slightly positive bits? Because the nucleus of the atom's kind of bare, because the covalent bonds, the electrons are at one side of the hydrogen atom, so its nucleus is kind of laid bare and slightly more positive because it's got a proton in. Why is the other side slightly more negative? Because it has electrons that are not used up in them covalent bonds. Right. Transpiration happens in the xylem, and it's really, really important to understand the adaptations of the xylem. So that's this next bit. So if you write notes on this, adaptations of the xylem. Right. Plant cells, I hope you understand now, have a cell membrane around the outside and then a cell wall going around the outside of that. They've got cytoplasm in the middle, and they also have a vacuole in the middle. Right, as a plant's roots grow or a plant stem grows, it makes new xylem cells, but eventually as these mature, the cell basically dies. So it makes a cell wall. The cell inside dies and leaves that really strong cell wall. So it becomes just a straw going all the way from the roots, all the way up the stem, all the way across the branches, into the leaves, and then you would have seen them little veins inside of leaves. That's xylem as well. Right, so the adaptations, I'm sure you'll be able to read this yourself, but it's got these really thick walls which are made of cellulose, and then also this other chemical called lignin. So if you want to push yourselves, like I've told you, you should probably be making a glossary. Hopefully you understand that some of the things in this topic, they're not necessarily difficult. It's just tons and tons of keywords to remember. So the cell wall is really tough. Why is it tough? Because it's made of cellulose and this new chemical called lignin. Why does it need to be? Because it's basically a straw, and if that straw collapsed, water wouldn't be able to get through and the top of the plant would die. Why is water so important? Because it's one of the reactants in photosynthesis. The plant needs it, otherwise it would wilt. So if a plant lost too much water, the vacuole would basically lose its water. The pressure inside of the cell would go down, so the plant wilts. Right, that's one thing. So it has a really strong cell wall, and that allows the plant to stand upright. Second adaptation is, so the cells grow when they're alive, then they die and they leave behind that in the cytoplasm, and the cell membrane vanishes. So the dead cells have no cytoplasm, so it basically, you can think of the xylem as just these big tubes. And then the cell wall between the cells, that also disappears. So what happens is that disappears, and one cell leads onto one cell, so it creates this big straw thing. So they're the adaptations of the xylem. There's another fact there, is that it's got tiny pores in the cell walls to allow things through, so things can get through without having to do osmosis and things like that, or active transport. They can just move through these pores through diffusion. Right, so we're kind of starting to put this together now. So water's sticky, the xylem's a big tube, and what translocation is, so a big overview, is it's how water moves through a plant. Water goes in through the roots and it goes out through the leaves at the top. Okay, so this is a diagram which explains transpiration, and you can go back to the start of the video if you want to see that definition again. So it's a movement of water through a plant, in through the roots and out through the leaf. Right, this is my explanation of transpiration. So remember, water is sticky. So water evaporates out of the stomata here. So basically what you can think of is all of these molecules of water are kind of one continuous stream. Because it's sticky, you can think of them all kind of sticking together. So when that one water molecule evaporates from the leaf, another one gets sucked into its place because of that the attractions between it. So one flies off, but the next one kind of moves up. So this would move up that way, and the next one would move up into its place, and the next one would move into its place, because they're kind of like little magnets. And that has an effect all the way down the stem into the roots, and that sucks water in from the soil. So transpiration is this cycle of water evaporating and sucking in new water from the roots. So every water molecule being lost from the leaves causes water to kind of move through the xylem, and it gets water pulled in through the soil, and it pulls water up the stem. So water gets pulled up because it is a sticky molecule, and that's transpiration. So We'll do a summary in the revision lesson. So tomorrow's lesson, is a revision lesson, and we'll do like a summary table explaining like where this happens, why it happens, what tubes it happens in. But it happens in the xylem. There we go. Right, one more thing about transpiration is how we measure it. So we could do an experiment in the lab to show that this is a thing and to measure the rate of it. So different things affect how fast this is happening. And one thing, so if you have like lots of warm, dry air, that's going to evaporate water quicker. So transpiration would happen faster. It might happen quicker at daytime than nighttime because the stomatas are closed at nighttime. But we can measure transpiration. So the reasons why we measure it, there's many different reasons why we would want to measure it and why it would change in different conditions. But this is how we measure it. Right, we use a piece of equipment called a pitometer, which is here. And what we'll do is we'll get a plant, and this would need to be airtight. So basically it would also be watertight. So any water that the plant's getting is going straight through the plant, through the stem, and none of it's going to kind of evaporate out the sides there. It's all going to go through the plant. Right, the next important thing in this is that we'll have an air bubble. So we would introduce a bubble into the end of this capillary, and what happens is, so as water evaporates, remember it's pulling the water through the capillaries, through the xylem, but in this case it's pulling water through this thing called a capillary tube, and we can measure how fast the bubble moves through that tube, and we can use that to measure the speed of transpiration. So what it says here is like millimeters per minute, so how many millimeters it would move through here. So we'd need a ruler. We'd put a ruler here and would physically measure how far the bubble's moving in a certain amount of time. So we could measure rate like that. Another thing I have seen in an exam was how much water has been taken up. So if we knew how many millimeters the air bubble had moved, and we knew the radius of this tube, so if we knew the radius was 2 millimeters, we could do pi r squared to work out the area of a circle where the radius would be 2 millimeters. So we did pi times 2 squared, so it would be 4 pi will be the area of that circle, and then we've times it by the length. So what we've worked out there is the area of that circle times it by the length. We'll get a volume because it would be 3D now. So you could use that to actually physically work out the volume of water that has moved through the plant, and therefore the volume of water that has transpiration has happened. So this is the equipment needed to measure transpiration. There's good free science lesson videos on pitometers if you'd prefer to have a look at them. Right, that's transpiration. The next thing we need to understand is translocation, and I think this is actually a little bit more complicated. So this is now translocation. Right, what moves in translocation? So it's sugar and mainly sucrose. So where in the plant, have a think about this, where in the plant would have more sugar? Would it be the leaves? Would it be the roots? So a good answer probably is the leaves, because the leaves are making sugar through photosynthesis. So quite often the leaves have more sugar than the roots. Can the roots ever have more sugar than the leaves? Yes. So in winter, plants like potatoes, in the summer they basically take all the sugar from the leaves and they store it in the roots, and that's where we get root vegetables from. So things like potatoes and carrots, that's a lot of stored sugar are in them roots. In the winter or in the springtime, they take them sugars and they pump them up the plant back into the leaves, so they can make new leaves, so they can make new leaf cells, so they can make new shoots and things like that. So it's not always from the leaf to the root, and it's not always from the root to the leaf, but what it is always from and to is a thing called a source cell. So that's where there's more sugar, and there's a thing called a sink cell where the sugar is going to end up. So where we want the sugar to go. And when I say where we want it to go in this question, it's where like we think it's going to end up. So obviously the plant's controlling this, but yeah, so it's not always from the leaf to the root, but in this example it is from the leaf to the root. But I can get rid of that if you want. So it does say leaf to root there, but it's from the source cell to the sink cell. Right, what's happening in translocation? So we've got tons of sugar there, and we need to move it down to the root. What happens is, so it moves through this type of cell called the phloem. So we'll have a look at what the phloem looks like now. Right, the phloem is fairly similar to the xylem. So it has these things in between the cells called sieves, which you've got holes in. So basically sugar and water can just move straight through from one cell to the other. It doesn't have to do active transport, it doesn't have to do osmosis, it can move through the cells. Right, how does sugar get into the phloem? So what happens is there's these cells at the site called companion cells, and they've got tons of mitochondria in, and what they do is they pump sugar into the phloem. So this is active transport happens here, and they are pumping sugar into the phloem. Right, so now what we've got is lots of sugar, lots of sucrose there. If you've got lots of sucrose, then that is naturally going to draw water in. Why is water going to get drawn in? So that's osmosis. So what happens first is active transport. Step 2, water moves in through osmosis. And the third thing that happens is that, so there's tons of pressure in there now. There was lots of sugar, that's drawn water in, and that's like, there's lots of stuff there now, so the pressure has gone up. So step 3, there's high pressure. So this moves all of that solution down through the phloem. When it gets to where it wants to go, where there's less sugar, then the sugar will either diffuse into the sink cells or active transport can happen. And the sucrose in that cell can then get made into starch. So potatoes store sugar not as sucrose but as starch. But that's translocation. So I think the best way to kind of understand that is a step-by-step thing of what's happening. So first, sugar gets pumped into the phloem. Next, water joins it because it's a really sugary solution. So water moves in through osmosis. And then third thing that happens is the pressure increases, so it forces that sugar down, and then it kind of gets removed into the sink cells where it can get stored. Right, this is the textbook version of the phloem. So I would pause that, just make sure you understand this now. So companion cells have got lots of mitochondria because they do active transport. This is a good diagram to kind of see the sieve cells.